Hello and welcome to this video in the series on integrated disease management in cereals. And in this video we're going to think about prediction of disease. So let's just manage expectation a bit, because I don't have a crystal ball and neither does anybody else to be able to say with certainty what the disease severity is going to be in this crop behind me later on in the season. But the more we understand about the epidemiology of the diseases, the better placed we are to understand what determines epidemics, to assess the disease risk, and from that to be able to make treatment decisions that are according to the need of the particular crop. So let's get started. Hello. This is a video in the series on the science and practice of integrated pest management, focused on disease control in cereals. And we're going to focus on the question how to predict epidemic severity using the example of Septoria tritici blotch in wheat. So why do we need to predict severity? And the answer is that it's an important principle of IPM that we treat according to need, and the need for treatment relates to the severity of disease during the yield forming period. So what we've got here are data from the Defra Pest and Disease survey since 1990, where we've been out to a couple of hundred winter wheat crops each year, measured the amount of Septoria in this case during grain filling, and also gathered data on the number of fungicide sprays applied in the bottom chart. So when I first became a pathologist back in the 1980s, we used to spray wheat once, then we sprayed it with fungicide twice, then we sprayed it with fungicide 3 times, and it looked for a while as if we were heading towards spraying it 4 times on average, but then we got a grip on the situation and it stabilised. Although it's fair to say that when we were spraying once, we were applying a full label dose, and over the years we've moved to a position of spraying between half and 3/4 of the label dose typically. But nevertheless, there are a couple of key points that come out of this analysis, comparing the number of fungicide sprays applied along the bottom with the severity of Septoria in the chart at the top. So the first thing is that it's hard to argue that we've been obtaining better control with the increased fungicide applications, and that's to a degree related to fungicide resistance issues, which we'll cover in a later video. But the other point is that we're not adjusting our fungicides according to the degree of risk. So if we look at years like 2020, when there was virtually no Septoria, or we look at years like 2012 or 2014, when there was a lot, fungicide treatments only adjusted to a modest extent. Now, Septoria isn't the only driver of fungicide treatment, but it is the predominant one. But the point is, this analysis is with the benefit of 2020 hindsight. We know from these data exactly what happened, but we're making these treatment decisions well ahead of knowing what the disease is going to do. So we're all in the business of prediction. And this is a pretty typical scenario. This is growth stage 39, the flag leaf's just emerged. It's a key decision point for fungicide treatment, how much to apply and of what. And at that point, for reasons we'll come to in a moment, the upper leaves of the canopy will always look clean. And what we're trying to do is make a prediction. If we do nothing, is the crop going to end up looking like the one on the left, in which case we need to do a lot, or is it going to end up looking like the crop on the right, in which case we may not need to do so much. So life is all about making decisions about whether to act or not, or what actions to take, and we all have a kind of mental model of the world and how it works that helps us make those decisions. So what I'm going to do through is what the mechanisms are that we should have in our mind as that mental model to help us decide on the risk of what's likely to happen in future. So this is the process that we're trying to manage. We have spores arriving on the leaf surface, germinating and infecting. We have then a period of symptomless growth, and the lattice of sort of purple fine lines here are Septoria tritici growing in between the cells of the leaf with no symptoms on the surface at this stage. In the next slide we have symptom expression, and at the same time as that we have the start of sporulation. And underneath the leaf surface there's a whole series of flask-like structures producing spores, and when the weather is humid those spores are exuded out through the leaf surface in a sort of sugary matrix, which can then be hit by rain droplets or surface moisture and spread back to other previously uninfected leaf surface. So that's the kind of biological process, but we need to be trying to think about this in quantitative terms. How much disease are we going to have? So let's think about this sort of quantitatively. On the left-hand side we have new leaves growing, producing green leaf tissue. If they don't get infected, those leaves will eventually senesce and die. If they do get infected, then part of that leaf area is going to become latent infections. And we can think about this in kind of chunks of leaf surface, the size of a lesion. And what we're interested in is, well, how many of those chunks of leaf surface are going to become latent infections that will subsequently become infectious, produce spores that go round and produce more infections on new green area. So the key things that determine the outcome are R0, which is the basic reproductive number, which is the number of daughter lesions produced by one latent lesion when that lesion is not competing with loads of other disease for leaf area. And we heard, unfortunately, an awful lot about this idea of R during the COVID pandemic, and it's the same principle here. And the other key determinant of the future epidemic is the generation time, which is the time for an infection cycle from lesion to lesion. And obviously the faster the pathogen goes through that generation time, the faster the epidemic can develop, which is part of the reason why potato blight is so incredibly damaging. But also Septoria has a high basic reproductive number and is also therefore capable of severe damage. And R0 is determined by the probability to survive to the infectious stage of a latent lesion, the number of spores produced, the probability a spore lands on a leaf, and the probability that spore infects the leaf. So what are the things that affect R0 and generation time? So three of the key determinants are here. So weather affects R0 and generation time. Variety and host resistance against disease affects R0 and generation time. And fungicide affects R0 and generation time. So there's a repeating pattern here. All of these things are largely kind of interchangeable. If we have weather that's less conducive, if we have a variety that's more resistant, then we need less fungicide, or vice versa. These things are all interchangeable and sort of interplay. However, to make the situation a bit more complicated, we also have the dynamics of the canopy growing. And that also interacts with the growth of the disease epidemic. So if we imagine we're at second node, the stem's started to extend, what will eventually become the leaf, the third leaf down the canopy has just emerged. And now it's emerged, it's exposed to spores arriving from elsewhere in the canopy, but there's currently no visible disease. We go on to third node, about 10 to 14 days later, another leaf has come out. There are still no sign of symptoms on the third leaf, and we're now getting infections potentially on the second leaf, depending on the weather. And then we get to flag leaf, 10 to 14 days later, another leaf has come out, and we just start to see the first lesions on leaf 3 lower down the canopy. So what that can look like in the field is shown in this image here. This is actually taken about growth stage 37, so the flag leaf is just that tiny spike in the right hand white oval. But this is a kind of worst case scenario that at that stage we're seeing lesions on leaf 4, which is in the left hand white circle. But if you look at the position of those lesions, which are producing spores at this stage, they're absolutely adjacent to and level with the emerging leaf 1 and the emerged leaf 2 and 3. So it doesn't need much rainfall, it only needs a bit of surface moisture and a bit of movement by wind in the crop canopy to spread those spores across between those leaves. So this is the worst case, but there's quite a bit of variation in what we see at this point. So these images were taken in 2 plots of wheat breeding lines that varied in architecture to some degree and also in disease resistance. And it was taken at growth stage 39 when the flag leaves just emerged. We need to look very closely here. If we look at the left-hand image, the highest Septoria lesions are in that white circle down the canopy, whereas the flag leaf is emerging somewhere near the top dotted line, and there's about 15 centimeters of separation vertically between those two. And the efficiency of dispersal goes down about 5-fold for each 10 centimeters of vertical distance. So the ability of those spores to reach those upper leaves, is really going to require some quite intense rainfall to achieve that, and the number of spores arriving is going to be lower. If we contrast that with the right-hand case, which was taken on the same day but in a different plot, we can see we've actually got sporulating lesions right up level with the emerging flag leaf, and the risk in that case is far higher. So we can think about a kind of short-term disease prediction about what's going to happen next. So if we think about the interaction between where the lesions are in the canopy and how much rainfall it's likely to take in order to move those spores to the upper leaves, we can get some kind of handle on what the future disease is likely to be like, and also take into account the disease resistance of the variety. Now, it would be nice to tell you that there was some neat algorithm that's been well validated for the UK to do that for you, but unfortunately there isn't yet, which is a shame. We can also make a prediction about how long it's likely to take for those lesions to appear, because the relationship between infection and lesion appearance and temperature has been well quantified, and that's in the chart on the right. So we can make a kind of reasonable judgment about the future progress of the disease in the short term. In the background to that, we can also take into account what's led up to that point. And surprisingly, there is also a relationship between the weather that occurs during the winter and through into the spring, leading up to the start of stem extension at growth stage 31, and the amount and severity of disease that occurs during grain filling. So in the paper cited at the bottom, what we showed was that mild winter temperatures and more accumulated days with greater than 3 millimeters of rain over the periods indicated by the horizontal lines, the combination of those two things is a useful determinant of the sort of background risk of disease that's then modified by the rainfall that occurs or doesn't occur from stem extension through to the emergence of the flag leaf and the ear. Now, that's a very long-term prediction, and clearly it's not going to be superbly accurate, but is it at all useful? And indeed, is disease prediction at all useful? So we took a look at this in a series of 44 field experiments where we grew resistant varieties and susceptible varieties to Septoria across a range of sites across the UK and across a range of seasons. And we could measure in untreated plots the Septoria severity at mid-grain filling. If we look at the top chart, what we can see here is the disease severity percentage along the bottom and the number of sites and seasons, in other words, the number of experiments in which that severity occurred. So for a resistant variety in that range of sites and seasons, in the majority of cases, the disease was less than 5% on the upper leaves, but there were also cases of much more severe epidemics, even on the resistant variety. If we then contrast that to the susceptible variety in the chart at the bottom, you can see there were rather few cases where disease was not damaging and many more cases of more severe epidemics. Now, for those 44 experiments, we were able to use the weather data to use that long-term disease prediction that I showed earlier to separate out these sites into sites for which we predicted disease would be low, or sites where we predicted there was a risk of a damaging epidemic. And if we look at that data, let's just look at the top row first for the resistant varieties. So there were relatively few of those sites where we predicted that disease would be low, but we were actually correct that at all those sites disease was low and there were no damaging epidemics. For those sites that we predicted the risk of a damaging epidemic, we can see that in fact there were some cases where there wasn't a damaging epidemic, but also cases where there were. So the prediction is not perfect. But the important thing was there were relatively few false negatives where on a resistant variety we predicted the absence of a damaging epidemic, but there in fact was. Whereas on the susceptible variety, the prediction was more difficult. So again, there was a preponderance of where we predicted low epidemics, that epidemics were in fact low, but also a few examples of those false negative cases where we predicted a low epidemic, but in fact it turned out to be damaging. So what we can see from this is that disease resistance in varieties is not perfect. Weather-related disease prediction is also not perfect, but the combination of the two is actually quite useful to help us manage the risk, because the difference between a resistant variety where a low epidemic was predicted or a susceptible variety where we were predicting a damaging epidemic, it is a pretty strong difference, and that can help us manage the risk. So what can we conclude about our ability to predict? So first of all, we can't make disease control decisions with the benefit of hindsight. They need to be made before the control is needed. So we are unfortunately in the business of holding a crystal ball. Epidemics are determined mainly by the basic reproductive number, R0, and the latent period, which determines the generation time. Weather, agronomy, crop genetics, and fungicides all affect the number of progeny that determines R0 and the latent period. Hence, disease risk and disease control variables are interchangeable. More control from agronomy and genetics or less conducive weather mean we need less fungicide. And for Septoria, the separation distance vertically between lesions and emerging leaves is a really good indicator of risk. And finally, the combination of disease-resistant varieties and weather-based disease forecasting, even if that forecast is in your head, this limits the range of future disease severities that are likely to occur and hence limits the risk. So thank you for watching.